Good afternoon. I'm Utsav Sharma, Product Manager for ATCC's Oncology Portfolio. Thank you for joining us in the ATCC's Excellence in Research webinar series. Today's webinar is titled Immune Checkpoint Reporter Cell Lines for Cancer Immunotherapy and Drug Screening. Today's webinar is presented by ATCC R&D scientists, Dr. Haiyun Chang and Dr. Kevin Tayo, who will be discussing our offerings of immune checkpoint reporter cell lines and their applications to streamline checkpoint inhibitor screening. Before we begin, a brief overview about ATCC. Founded in 1925, ATCC is a nonprofit organization headquartered in Manassas, Virginia, with an R&D and services center in Gaithersburg, Maryland. We are one of the largest and most diverse biological materials and information resource for cell biology, often referred to as the gold standard when it comes to authenticated cell materials. We are an innovative R&D company that is focused on developing new and advanced cell models and tools to accelerate oncology, immuno-oncology, drug discovery, and screening. With that, I'd like to briefly take the opportunity to showcase our oncology portfolio of products broken down by application here. At ATCC, we offer classical cell products in the form of human and animal cell lines, certified reference materials, as well as other cell culture media and reagents. For biomarker discovery, we have matched tumor and normal cell lines, quantitative cell line DNA, as well as cancer cell panels. We support tumor biology research by a collection of reporter-labeled cell lines and cell lines broken down by gene mutations. For drug screening applications, we have a multitude of primary cells from various tissue types at low passages, CRISPR-engineered isogenic cell lines, as well as a robust cell immortalization program. For immuno-oncology applications, we have our classical portfolio of primary immune cells and cell lines, hybridomas for antibody manufacture, THP1 monocyte cell line in various reporter formats, as well as our newest offering of CAR-T target and checkpoint reporter cells that you'll be hearing about today in today's webinar. Finally, we have a growing collection of patient-derived models offered in collaboration with the Human Cancer Models Initiative, or the HCMI, in the form of organoids, adherent and suspension cells, as well as conditionally reprogrammed cells that we have companion growth kits and exhaustive cell culture guides for easy adoption into immuno-oncology platforms. In today's webinar, our scientists will provide a brief overview of the field of immuno-oncology, share data on protein profiling of the ATCC cell lines, and provide insights into application data that we've generated with the checkpoint reporter cell lines. Followed by will be a functional comparisons of the modified and parental cell line. At the end of the webinar, there'll be a Q&A session. Please use the chat function to drop in any questions you may have during our session, and they'll be answered at the end of the webinar during the Q&A session. With that, I'll hand over to Dr. Haiyun Chang.
Thank you, Utsav, for the introduction. Good afternoon, everyone. My name is Haiyun Chang, and I'm a scientist at ATCC. Today, I'm going to introduce you to ATCC's newly launched immune checkpoint reporter cell lines and explain why and how we developed these cell lines. Immune checkpoint molecules are the markers on the cell surface that communicate self to the immune cells. When T cells find these markers on the surface of another cell, they recognize the cell expressing these markers as self and do not attack. Cancer cells use this to their advantage and increase the expression of these markers to evade immune detection. Scientists have found and studied quite a few of these immune checkpoint molecules, and examples of them are shown in the picture on the left. Of these molecules, the reporter cell lines that we will be introducing today endogenously express high levels of PD-L1, CD155, and B7H3. These markers are important in cancer research because we can develop therapeutic antibodies against them and block the interaction to initiate T-cell mediated immune responses in the tumor microenvironment. The images on the right show you an example of how these immune checkpoint inhibitors work. Therapeutic antibodies against PD-L1 and PD-1 bind each target molecule on the cancer cell and T cell respectively and prevent the immune checkpoint recognition. This leads to the activation of the T cell to begin the killing of the cancer cell. The history of these immune checkpoint inhibitors is short, but they have become a significant breakthrough in the field of cancer immunotherapy. I will talk about the history of immune checkpoint inhibitors briefly in the next slide. The first immune checkpoint molecule was discovered in 1987 and named cytotoxic T lymphocyte antigen number 4 or CTLA4. However, the function of this molecule was not clearly understood until 1995 when Dr. James Allison and his team identified it as a crucial immune checkpoint molecule with great potential as a future anti-cancer therapy target. The first CTLA4 blocking antibody was immediately developed and tested on animals a year later in 1996. Soon after, programmed cell death 1 or PD-1 was identified as another immune checkpoint receptor expressed on T cells. The first checkpoint inhibitor approved by the FDA was ipilimumab, a CTLA antibody in 2011 for the therapy of advanced melanoma. Subsequently, two PD-1 antibodies, pembrolizumab and nivolumab, obtained FDA approval in 2014. Since then, the FDA has approved 9 checkpoint inhibitors that target the CTLA4, PD-1, PD-L1, and LAG3 pathways for the treatment of more than a dozen major cancer types. In 2018, Drs. James Allison and Tasuku Honjo were awarded Nobel Prize in Physiology and Medicine for their pioneering work on checkpoint molecules CTLA4 and PD-1 respectively. Even after about 2 decades of explosive growth, however, considerable challenges remain in development of immune checkpoint inhibitors. One of the major challenges is the intrinsic complexity of cancer immunology study models. Currently in the market, there are in vitro artificial systems available comprised of cancer cells that are engineered to overexpress certain immune checkpoint molecules and non-cytotoxic T cells that are engineered to express T cell activation signaling reporter genes. These in vitro systems are relatively easy to use and the results are generally reproducible, but since these cells are artificially engineered, the subsequent intracellular events following immune checkpoint inhibitor-induced immune activation are missing from the system, which is crucial in maintaining the physiological relevance of the system. Another frequently used model type is syngeneic or humanized mouse models. These are classic immunology models that enable us to understand the immunological mechanisms as a whole and test potential therapeutics. However, the interspecies disparity between the mouse and the human immune system is inevitable. The fully human ex vivo assay models that utilize human T cells and human cancer cells solve this issue. These are typically co-culture assay models of primary immune cells and patient samples that maintain physiological relevance, unlike the artificial models. However, the intrinsic donor variability issue makes it especially difficult to obtain reproducible results using these assay models. So what is the solution? A physiologically relevant assay system that can generate highly reproducible data would be the solution. So at ATCC, we have developed the immune checkpoint reporter cell lines that endogenously express high levels of immune checkpoint molecules. These reporter cell lines provide the physiological relevance that is more relevant and more predictive to the in vivo events, unlike the currently available artificial in vitro assay models. We have also achieved high reproducibility with these reporter cell lines by utilizing immortalized cell lines that eliminate the donor variability issue that is intrinsic to the donor-dependent human ex vivo assay systems. And by inserting Luciferase reporter gene, the cell lines allow convenient data collection and enable reliable measurement of the potency and stability of immune checkpoint inhibitors. And through the next set of slides, I will explain more in detail how we developed the immune checkpoint reporter cell lines. For physiological relevance, we first wanted to find the cell lines that naturally express high levels of immune checkpoint proteins. We performed protein profiling by flow cytometry of over 50 cancer cell lines of various origin available at ATCC. The cell lines were profiled for both inhibitory and co-stimulatory immune checkpoint molecule ligands in the presence and absence of interferon gamma, a cytotoxic cytokine released by the immune cells. A heat map was generated based on the median fluorescence intensity, and each column was color-coded separately to avoid cross-comparison among different antibodies. The data here show which cell lines endogenously express which immune checkpoint molecules, and we selected our candidate reporter cell lines based on this information. As highlighted in the table, the 3 immune checkpoint reporter cell lines that we're going to introduce to you today were developed from MG63, a breast cancer cell line with high expression of CD155, a TIGIT ligand, NCI-H1650, a lung cancer cell line, which expresses a high level of B7H3, and HCC827, another lung cancer cell line that endogenously expresses a high level of PDL1. After identifying the cell lines that endogenously express high levels of checkpoint molecules, we devised a reporter assay system that utilizes a luciferase reporter gene for an easy, rapid, and sensitive quantification of the target gene expression. The lentigas-Luc2P plasmid contains a luciferase reporter gene under the control of gamma interferon activation site or GAS response element. So when the interferon gamma is released from the immune cells and activates the JAK-STAT signaling pathway, it would induce the transcription of the luciferase reporter gene. After the viral transduction of the plasmid into the selected cell lines, the cells were then enriched by puromycin selection, and single cells were isolated by automatic cell sorting. The single cell clones were expanded and evaluated by interferon gamma stimulation. The clone that yielded the brightest luminescence upon interferon gamma stimulation was selected to be developed into the immune checkpoint reporter cell line. Here, this picture shows the mechanism of action for the immune checkpoint reporter cell lines. Normally, an immune checkpoint ligand expressed on the surface of a cancer cell, such as PDL1, would bind a complementary immune checkpoint receptor on the surface of a T cell and inhibit the activation of the T cell. When the binding is disrupted by an immune checkpoint inhibitor, such as a PD-1, PD-L1 blocking antibody, the T cell gets activated and releases various cytokines, including interferon gamma. The released interferon gamma is then recognized by the interferon gamma receptor on the surface of the cancer cell, and this leads to the activation of the intracellular JAK-STAT signaling pathway. The JAK-STAT signaling pathway activation induces the interferon gamma activation vector binding to GAS promoter, and in our system, this promotes the GAS-induced transcription of the luciferase gene. The series of events produces easily detectable bioluminescent signal that enables reliable measurement of the potency and the stability of the immune checkpoint inhibitor. Now, Dr. Kevin Tayo will come up and present the data we have generated using these immune checkpoint reporter cell lines.
Thank you, Hyun, And good day, everyone. My name is Kevin Tayo, a scientist at ATCC. Before I dive into our functional data, I would like to take some time to talk about our work in ensuring that these reporter cell lines maintain physiological relevance to their original parental counterparts. Here, we wanted to ensure that the physical properties of the unmodified parental cells are preserved in our gas loop 2 modified reporter cell lines following viral transduction. Here, we show representative microscopy images of some of our reporter cell candidates. At the top, we show representative images of parental HCC 827, MG63, and H1650 cell lines, as well as their gas loop 2 modified counterparts shown below. As you can see, the morphology of the modified cell reporters exhibit no discernible physical differences compared to their parental counterparts. Next, we compare the growth rates and cell doubling times between parental and gas loop 2 modified cells. For this study, we incubated cells for 5 to 7 days and assessed changes in the cell numbers daily. These graphs illustrate the daily increase in cell number for each cell line during their designated incubation time. Using this data, we were able to calculate the doubling time for each cell and compare differences in growth rates between parental and modified cells. Here, we show that all three reporter cell lines exhibit similar growth rates relative to their parental cell analogs, all showing changes in growth rates well below 20%. Additional studies were conducted to ensure that the high target expression in each monoclonal gas loop 2 cell line was comparable to their parental equivalents. Here, flow cytometry analysis was performed to compare the expression of PD-L1, CD155, and B7H3 in HCC827, MG63, and H1650 cell lines, respectively. These graphs illustrate the expression of the respective target, shown in red, as well as an isotype control, shown in blue. Expression of the parental cells are shown on top, whereas their gas loop 2 modified cell counterparts are shown below. For all three reporter cell lines, the target expression was shown to be comparable to their unmodified parental cell lines. Additional flow cytometry studies were conducted to ensure that the target expression in modified cells would be maintained following prolonged cell culturing. Here, we compare the respective target expression in low passage number cells, shown on top, to cells cultured above 30 population doubling levels, or PDL, shown at the bottom. Here, we show that high expression is maintained in all three cells, even after prolonged culturing. Next, I want to switch gears and discuss some of the application data we generated, showcasing some of the functional studies of these modified cell lines. So following the gas loop 2 plasmid transfection of our candidate cell lines, we wanted to examine which cell line for each target would provide the highest change in luminescent signaling from the interferon gamma stimulation. Here, we show the results of a series of interferon gamma stimulation assays of candidate immune checkpoint reporter cell lines for PD-L1, CD155, and B7H3. All cell lines here showed increased luminescent signaling upon stimulation with the increasing concentrations of interferon gamma. The cell lines that yielded the highest fold change relative to untreated controls were selected for the next set of experiments. For PD-L1 reporter cell lines, HCC827 gas loop 2 cells consistently showed the highest fold change in luminescent signaling following stimulation with various interferon gamma concentrations. Similarly, MG63 and H1650 cells were selected for CD155 and B7H3 respectively. After clonal selection, the three cell lines were once again tested with cytokine stimulation. All three cell lines demonstrated high sensitivity to 24-hour interferon gamma administration, especially the HCC827 gas loop 2 cells, which exhibited an average of 50-fold increase in luminescence relative to untreated controls following a 0.01 nanograms per mil interferon gamma administration. Increased luminescence was observed in all cell lines with increasing interferon gamma administration, with 1,000 nanograms per mil interferon gamma administration resulting in a range of 125 to 250-fold increase in luminescence compared to untreated controls. Additional interferon gamma stimulation tests were conducted on reporter cells with high PDLs to ensure that each reporter cell line maintained high luminescence signaling from stimulation even after extensive culturing periods. Here, we show that even after 30 population doubling levels, all 3 reporter cell lines maintain high luminescence signaling. Following the interferon gamma stimulation assays, the signaling activation was also assessed using conditioned media from primary human T cells. Here, the 3 cell lines were treated for 24 hours with conditioned media from either non-activated or anti-CD2, CD3, CD28 bead activated primary human CD8 cytotoxic T cells, followed by luminescence detection. The conditioned media from the activated CD8 cytotoxic T cells increased luminescence values by over 100-fold in all 3 cell lines, relative to media taken from non-activated T cells. Next, the ability of the cell lines to serve as a checkpoint inhibitor reporter was evaluated by primary human CD8 cytotoxic T cell co-culture studies. Here, monoclonal HCC 827 gas loop 2 cells were co-cultured with different ratios of CD8 cytotoxic T cells, as well as under different co-culturing times. The cells were also administered with various concentrations of either a PD-L1 monoclonal antibody or an isotype control IgG1. Increasing the PD-L1 antibody concentrations in the co-culture models resulted in a dose-dependent increase in the luciferase expression, exhibiting a 3- and 5-fold increase of luminescence signaling relative to untreated controls under the conditions of a 24 and 2-hour co-culture incubation period with a 1:1 and 1:10 ratios of HCC 827 gas loop 2 to CD8 T cells, respectively. In contrast, increasing isotype control IgG1 concentrations failed to induce any luminescent signaling from HCC 827 gas loop 2 cells relative to untreated controls. Co-culture studies were also conducted on MG63 gas loop 2 reporter cells. This time, cells were cultured with CD4 helper T cells at 24 and 48-hour incubation periods. Here, cells were administered various concentrations of either a TIGIT monoclonal antibody or an isotype control. Again, we observed that increasing TIGIT antibody concentrations in the co-culture models results in a dose-dependent increasing of luminescence intensity. Here, luminescence signaling was 3- and 6-fold larger than untreated controls for a 24 and 48-hour incubation times, respectively. Again, there was no observed increase in luminescence intensity from increasing concentrations of the isotype control from treated cells relative to untreated controls. Finally, co-culture studies were conducted on H1650 cells using natural killer cells at a 2:1 ratio. Incubation times for these co-culture studies were conducted at either 24 or 48 hours. Here, co-culture models were administered with increased concentrations of either a B7H3 antibody or IgG controls. Again, increasing the administered concentrations of B7H3 antibody in the co-culture models results in a dose-dependent increasing of luminescence intensity, with a 2- and 3-fold increase luminescence signaling relative to untreated controls at 24 and 48-hour incubation times, respectively. Yet again, there was no observed increase in luminescence intensity from increasing the concentrations of the isotype control relative to untreated controls. Now, let's summarize our findings. The recent success in the use of immune checkpoint inhibitors in the treatment of cancer shows enormous promise. However, new tools and models in the field of immuno-oncology are desperately needed. To serve this urgent need, ATCC conducted an extensive protein profiling of our substantial catalog of cancer cell lines, T cell lines, and primary T cells to assess for high expression of selected immune checkpoint molecules and their ligands. Based on this protein profiling data, we have generated and developed several immune checkpoint reporter cancer cell lines for use in the convenient detection of immune checkpoint blockade-induced signaling. Here, we showcase 3 cell lines that provide stable and robust luciferase signaling produced by relevant stimulation, enabling reliable measurement of the potency and stability of immune checkpoint inhibitors. The endogenous high expression of PD-L1, CD155, and B7H3 and HCC827 gas-Luke2, MG63 gas-Luke2, and H1615 gas-Luke2 reporter cell lines, respectively, delivers physiological relevance to the immune checkpoint assay. For more information on this topic, please visit us at atcc.org/immuno-oncology.
Thank you, Hayun and Kevin, for the insightful presentation. In just a few minutes, we will begin our Q&A session. At this time, please use the chat function available through the webinar program to submit any questions. The recorded webinar presentation will be available on demand on the ATCC website at www.atcc.org.
Thank you, everyone who's posted in questions already through our chat function. At this time, please use the chat function to enter any questions that you may have. We've had a bunch of questions come in already. This first question, maybe for Kevin, what is the passage limit of the immune checkpoint reporter tumor cell line?
Yeah, that's a great question. We had tested and confirmed the ability of these cell lines up to above 30 population doubling levels, but they can go beyond at least 60 and 90 population doubling levels. It's one of the great things about having these continuous cell lines is that I'm not going to say that they are going to work indefinitely, but they will work for a very long time, so long as that you have maintain appropriate culturing methodology. So don't let them become overly confluent, maintain appropriate cell density, as well as that you maintain appropriate media as well. Don't let things expire.
Great, thank you, Kevin. Here's the next one. Hayun, why don't you take this one? Is it necessary to use activated T cells for co-culture?
So activated T cells are good to use when you are testing the cells initially, because activated T cells, they release high levels of interferon gamma. I'm talking about CD8 positive T cells right now, or you can also use CD4 T cells that release interferon gamma. But when the T cells are activated, they will release high levels of interferon gamma, and it will elicit the luminescent signal from the reporter cell lines. However, if you're using these cell lines to test your immune checkpoint inhibitor, I would say that if you use the activated T cells, they will have high background signal. So and you want your immune checkpoint inhibitors to actually be activating the cells. So I would recommend that using not as activated T cells, so that you have low background levels, as well as you get high signals for your immune checkpoint inhibitors.
Great, thank you, Hayun. There's another question that's quite similar to this question. Do we have to use CD8 positive T cells for co-culture?
So it's not necessary to use strictly CD8 positive T cells for co-culture, because this luciferase expression is in response to the presence of interferon gamma released from immune cells. So any immune cells, for instance, like Th1 cells that can produce interferon gamma when activated, can be used to elicit the luminescent signal from the cells, from the reporter cells.
Great, thank you, Hayun. Another question we've gotten is about the application data. The question is, how can we get the access to the ATCC application data? So the ATCC application data, we publish that on our website by journal publications, conference abstracts, including this webinar where you saw all of the application data. It's freely available on the ATCC website, www.atcc.org. You'll find our resources button that lists all our current resources. If you have any specific questions about the application data, please contact ATCC customer service directly, and they can forward your very specific questions to us at the ATCC R&D. All right, so let's do another one. So this one's another one about the co-culture assay. So how did you do the co-culture with the checkpoint reporter T cells? Did you use CD8 positive T cells or CD4 positive T cells? What are your recommendations for optimizing co-culture conditions? Either Hayun or Kevin.
Let me take this question because I also see that a question came in that asks how long the results are valid after addition of the immune cells. And this question is also connected to the question that you just read with Seth. And so first of all, you can use either CD8 positive T cells or CD4 positive T cells. It depends on your need. But one thing to keep in mind, if you choose to use CD8 positive T cells, and this also connects to how long the results are valid, is that CD8 positive T cells are cytotoxic T cells. Once they're activated by the immune checkpoint blockade, they will eventually kill the tumor reporter cells. And because dead cells cannot express those freeze, you will not see the luminescent signal when the cells die. So you will need to optimize the time of co-culture so that you see the maximal signal increase due to the immune activation and subsequent interferon gamma release before you see the decrease of the luminescent signal due to the cell death. If you're choosing to use the CD4 positive T cells, then this is not a concern. But make sure. But we also saw that there is an increase, depending the initial increase, and then the signal will decrease a little bit and plateau. So make sure to check do the time point experiment first to see like which time point suits you the best, or which time point, because it will vary from experiment to experiment. And when you're using CD4 positive T cells, make sure to check that you're using the right subset of CD4 positive T cells that releases interferon gamma in response to immune checkpoint blockade, such as Th1 cells.
Great, thank you, Hayun. We have another question. This one is, if I want to screen PD-1 inhibitors, what are the cell lines from ATCC to be used for the assay?
So if you want to screen the PD-1 inhibitors, we have the HCC 827 gastric cell line that highly expresses PD-L1 immune checkpoint molecule. So you can use that cell line to screen PD-1 inhibitors.
And in addition to that, just to mention, at ATCC, we have a roadmap, and we'll be soon developing our PD-1 checkpoint inhibitor cell line in the form of a T cell line, and that will be available in fall of 2023. All right, there's another question on T cells again. Hayun or Kevin, how do we activate the T cells?
Kevin, I guess I could take that. Yeah, sure. So when we activate these are primary human T cells. So we activate them by using these are commercially available. They're anti-biotin bead particles that are loaded with biotinylated antibodies against human CD2, CD3, and CD28.
Great, thank you, Kevin. So we have another question about the application data. How can we get the protein profiling data for the immune checkpoint reporter cell lines? I guess I can answer that. Very similar to our application data, all of this is available on the ATCC product website for each of the cell lines. And you can find this relevant information in the section of characteristics. In addition to that, if you go to our resources page, we have a brochure that would list the application data as well as the protein profiling data you saw in our slides today. If you need any further information, please reach out to the ATCC customer service directly, and we'll be able to forward your questions to our R&D. So this next question is, how can we be sure that the cell lines work?
I guess I could take that. So this data will be available on our website. So you can check the, I think it should be under resources at www.atcc.org, and you can find the data for review.
And on the similar lines, Kevin, there is a question: how long are the results valid after addition of the immune cells?
So,
I think that'd be a great question for Ayun. Sorry, go ahead, Ayun.
So yeah, I kind of answered the question with my previous answer to the co-culture question. So so after you add the immune cells, you'll want to monitor the signal increase and decrease over time before you select the optimal time, the incubation time for the co-culture. It will vary from cell line to cell line, and also like what type of immune cells that you're using. What we normally use is that we go from as short as 2 hours, 4 hours, 6 hours, 8 hours, overnight, 24 hours, 48 hours, up to 72 hours. Usually we see the results either overnight or 24 hours. If the immune cells are reacting quickly, then we see the results as short as 2 hours. And if the immune cells are not activated as robustly, then we see the results 24 hours, 48 hours-ish usually. So that's the time frame that I would recommend, but you will probably want to monitor for your own experiment how the signal changes over time, and then select the optimal time point for your experiment.
Great, thank you, Ayun. So we have another question that says, are there any licensing requirements for this cell lines? Yes, there are ATCC licensing requirements for the cell lines. The full licensing and material transfer agreement requirements that we have for all of our cell lines are usually found under the permits and restrictions section on our product page on the website. And if you have any additional requirements that you would like to go into more detail about, please contact salespermits@atcc.org. All right, we have another question that just came in. Are there specific cell culture requirements for these cell lines?
Yeah, I could take that.
Yeah. Mm-hmm.
Yeah. So the answer is yes. Each cell line is going to have a specific requirement how to culture these cells. You can find this on the product information, on the product page or on the ATCC website, and we will provide you with full information on how to handle and subculture these cells. So we'll have protocols that will go through things like cryopreservation and media choice, details also of every product that you'll need to handle and utilize these cells. And so, by performing this, you will have hopefully straight sailing with the culturing handling of these cells.
Great, thank you, Kevin. Here's another one. I could not get the name of the breast cancer cell line that is available. Could you please state the name?
So we don't have this breast cancer reporter cell line at this moment. The cell lines that are available are HBC827 and NCI H1650 that are lung cancer cell lines, and we have MG63 gastric tissue cell line that is bone cancer cell line.
Okay, thank you, Ayun.
Mm-hmm.
Here's another question. How does ATCC do single cell cloning? And have these cell lines, a similar question we have, where have these cell lines been developed by single cell cloning?
Yes, the cell lines for...
Go ahead.
Yes, the cell lines were developed by single cell cloning, and we use an automated single cell sorter from Sony to do the cell sorting. So yes, they were developed by single cell cloning.
Great, thank you, Ayun.
Yeah.
Here's another question. Do you only have tumor cells or for immune checkpoint reporters? Will any immune cells be available?
So now we only have the 3 tumor reporter cell lines available on the ATCC website for you to order. But we are planning on expanding these immune checkpoint reporter cell line products to include more cancer types and cell types. So please look for any updates on ATCC website, or if you have a specific interest, please let us know and we can contact you, like respond to you individually.
Great. Thank you, Hayun.
Sure.
Here's another question. Can these cell lines be used for other checkpoints other than PD-L1, CD155, and TIGIT?
This is a great question. We have this profiling data, protein profiling data of the immune checkpoint molecules available on the ATCC website. And if you look at that data, it's also the table that we showed you before. These cell lines, the cell lines that we have available right now, we tested them for PD-L1, CD155, or TIGIT, and B7H3, but they also express high levels of some other checkpoint molecules as well. And you can use them. We have not tested them. We have not thoroughly tested these cell lines for these other immune checkpoint molecules that they highly express, as we did with PD-L1, CD155, or B7H3. But in theory, yes, they can be used to test other immune checkpoints that they endogenously express.
Great. Thank you, Hayun. Here's another great question. I guess I can take this one. What QC tests have been performed by ATCC on these cell lines? Our full list of the quality control tests we do, they're pretty standard. They're for each cell line under the detailed product information on the product page that you see for each of our cell lines on the website. In general, we perform full list of bacterial and fungal tests. We do mycoplasma testing, virus testing, and in addition to what functional tests we were described today in our webinar, we also fully SDR profile these cell lines. Okay, here's another question. What other tests were performed in comparing parental and modified cell lines?
Yeah, I can take that. Yeah, that's a great question. So just as a review, when we looked at the parental and modified cells, we compared their morphology, growth rates, and the checkpoint molecule expression. So this is the data we've shown. But in addition, to ensure that we actually have the cells that they actually are the same cells, we performed CO1 mitochondrial gene testing and short tandem repeat profiling to validate the authenticity of these modified cell lines. Moreover, we've done other quality testing to ensure, as Gustav had mentioned, that these materials are free from bacterial, fungal, or mycoplasma contamination. So the growth rate is indeed because this is literally the same cell that's been slightly modified. It's not a mycoplasma contaminated HeLa cell. So when we produce these products, you're going to get exactly what we provide. And we have authenticated this.
Great, thank you, Kevin. Oh, here's another great one. Do you have to use a PD-L1 antibody for the PD-L1 reporter cell line? Can you use the PD-1 antibody?
Yeah, I'll take this question. So
sure.
Yes, can either you can use either one. You can use PD-L1 or PD-1. In fact, for the CD155 reporter cell line, which is MG63 gastric cell line, CD155 is a ligand of TIGIT immune checkpoint receptor. So when we were generating our application data for the co-culture, we actually used TIGIT blocking antibody instead of CD155 antibody, and we got a great result. So yes, you can use either one. You can use PD-L1 or PD-1, whichever is the one that you're testing.
Great. Thank you, Hayun. So we have one final question here. Do these cell lines support bispecific checkpoint inhibitor screening? I guess I can sort of allude to that. We've profiled these cell lines completely for all of the common checkpoint molecule expressions, as you saw in our data today. We've also done some protein profiling on our co-stimulatory molecules. So, and that data is available on the ATCC website. So if you have a molecule that you're testing, which is a bispecific checkpoint inhibitor, they might support that if the cell line actually expresses that checkpoint. So you'd have to check with our profiling data. However, we at ATCC have not performed any screening for bispecific inhibitors. Great. At this time, if there are any additional questions, please use the chat function to drop any questions. So here's another question. Have you seen any difference between different available anti-PD-1 antibodies?
So let me take this question. We haven't tested many different antibodies to exclusively see the difference between blocking antibodies. So this is not from our own data, but then these reporter cell lines are developed to check the efficacy between how effective this antibody is in terms of blocking the immune checkpoint recognition. So I would assume that there would be differences between different antibodies. That being said, we have not tested different PD-1 or PD-L1 antibodies to compare these antibodies, like which one is more effective and which one is less efficacious. So what we're providing here is that you can test the efficacy using the reporter cell lines, but we haven't done those efficacy tests comparing different antibodies by ourselves.
Great, thank you, Hayun. Just as a reminder, if you have any additional questions, please drop them in the chat function. Now, I want to talk a few minutes as we are waiting for people to put in additional questions about some of the launch products that we have at ATCC for the immuno-oncology portfolio. As listed here in our slide, we have released previously last year 3 CAR-T target reporter cell lines can be used to check which have high endogenous expression of the CAR-T antigens and can be used to actually test CAR-T target potency and efficacy. These are available for CD19, CD20, and for solid tumor support in form of HER2. In addition to that, we've launched the PD-L1, CD155, B7H3, and on our roadmap, soon you'll be seeing a PD-1 reporter cell line coming out later this year with NFAT as the promoter of choice. In addition to our reporter cell lines and CAR-T target reporter label cell lines, we also have a vast collection of patient-derived models. These models are in collaboration with the Human Cancer Models Initiative. They come in diverse genetic backgrounds. We have culturing protocols as well as organoid growth kit support that you could be used for easy adoption of these HCMI models. We had a similar question in regards to the breast cancer cell line, Hayun. If you could please clarify. The question says, yes. Did you say one of the cell lines was the breast cancer cell line?
So the short answer is no. Unfortunately, we don't offer breast cell line, reporter cell line at this moment. We are planning on expanding to include more tumor types. But right now we have 3 cell lines, 1 bone cancer cell line and 2 lung cancer cell lines.
Great. Thank you, Hayun. If there are any additional questions, please feel free to drop them in the chat function. Just as a reminder, the webinar recording for today's session will be available on our website, www.atcc.org. And additional resources regarding all of the ATCC products would be available on our website. And in addition, you can use our website to contact our sales team to get in touch with our R&D if you have additional questions in regards to our cell lines that we discussed today. All right, with that, this marks the end of our webinar. Thank you again so much to all the participants for active participation in today's webinar. If you have any additional questions, please reach out. Thank you again so much. And thank you to our presenters, Hayun and Kevin, for an excellent presentation today.
Thank you, Sav.
Thank you.